thing. A virus is primarily made of a head and in some cases a tail. The head of a virus contains the capsid, or an envelope made of proteins, and the genetic material in the inner core. This genetic material can either be DNA or RNA. A relatively complex virus is called a bacteriophage.
What do bacteriophages do?
Bacteriophages are viruses that replicate within bacteria. During viral replication, virus particles attach to their cells and release genetic information within the host cells. When the cells are infected with this foreign genetic material, the enzymes of the host cells synthesize new virus particles. Once these new virus particles are assembled, the host cells go through lysis, or the bursting of cells. Lysis allows for the new virus particles to be freed.
But what happens to the material inside the host cell during lysis?
In marine settings, lysis of cells releases dissolved organic matter and particulate organic matter. This process is called viral shunt and drives carbon recycling in oceans.
Is the carbon cycle the only thing affected by viruses in marine ecosystems?
Actually, no. Scientists have previously focused on viruses' effects on exclusively the carbon cycle. However, viral shunt doesn't impact the carbon cycle as significantly as viral shunt affects the phosphorus and nitrogen cycles. This is because viruses, unlike living organisms, don't contain a relatively large concentration of carbon. Viruses are primarily composed of proteins, which contain carbon and nitrogen, and DNA or RNA, which contain carbon, nitrogen, and phosphorus. All this organic matter can be absorbed by marine microorganisms or pulled to the bottom of the ocean once released during lysis.
How much nitrogen and phosphorus is released from viral shunt in oceans?
Well, we don't know exact numbers, but scientists have made some mathematical models to help us estimate the number of base pairs within a given capsid, the number of capsid proteins, and the amount of material released following cell lysis.
I can't wrap my mind around this. Where is this happening?
Well, the magnitude of the effect of viral shunt on the value of dissolved organic phosphorus within oceans varies based on the virus density and dissolved organic phosphorus concentration. When virus density is over 3.5 to the 10th power virus particles per liter, and the dissolved organic phosphorus concentration is over 100 nanomolars, marine viruses can contribute greater than 5% of dissolved organic phosphorus concentration of oceans. These parameters were derived from studies conducted in various locations, including the Sargasso Sea and within the North Pacific subtropical gyre. The dissolved organic nitrogen content within oceans can also sometimes be affected by viral shunt.
If this process has been happening forever, why do we care so much?
Well, scientists are interested in viral shunt in oceans because the process has the potential to contribute significantly to global nutrient cycling. Scientists are realizing that the phosphorus content of virus particles is the predominant contributor to nutrient cycling, with nitrogen content playing some key roles, and the carbon content of virus particles minutely contributing to nutrient cycling.
One last question. How do you know so much about this?
Well, in my bio class, we've studied biogeochemical cycles and macromolecular structures.
Oh, okay. Well, thanks for the help.
No problem.